you know, you could probably make use for them if you want. Yeah, we don't need them.
Okay, I just want to make sure.
All right, get Kahoot pulled up for you. Anybody have any questions before we get started with the routine stuff? I know that it was a lot getting those concept maps done. You will, every other week, pretty much past the first week, you should only, normal weeks, we'll really only have 2. So that was the big hurdle. If you made it over that, and remember, you get your 2 lowest scores dropped also. So keep that in mind as well. So if you chose not to do 2 of them, that's your 2. Or if you chose not to do 1, you still got 1. It'll automatically drop those. And if you have 4 of them submitted already, it's dropped 2 of them anyway. Like seriously, it drops 2 of them anyway. So even if they're all high scores, so it drops 2 no matter what. If you're all high scores, then your 2 lowest scores are high scores. So, but that shouldn't be affecting your actual grade by dropping the high scores, just because remember, this is not a points-based class. It is a percentage-based class, right? All right, just be clear. And if you guys have any confusion about that, then just let me know, because you can play with your grade predictor calculator on Canvas, but if it's confusing to you for any reason, just let me know and I'll clarify that. At least every time, this is getting really annoying. I didn't used to have this problem at all. And it's been every single computer that I've gotten on, it does this. Get our Kahoot up. Okay. Lecture. This will be trying to find chapter 2. I think it's in service. All right, so we'll be starting with chapter 2, which is our chemistry stuff first. So we'll pull this up and then get us logged in. We got a minute or 2 for that. Four, 4, 6, 0, 7. Today will be our first day of business as it will be every time. There will never be a day when I won't call on you. So I mean that quite seriously. I meant that when I said it. It wasn't a joke when I said it. What I'm saying this and reminding you guys is that I was actually pretty surprised how terribly my first in-person class went. I'm hoping you guys will do a little bit better now that I'm kind of warning you ahead of time. I'm reminding you I wasn't joking when I said I would call on you, and I wasn't joking when I said I expect you to answer the question. Okay, so I'm not saying that they didn't answer the question. Most of them did pretty good, but I had definitely a few people in that class that were like, you know, you don't get points if you just sit there and say, I don't know, or if you don't know the answer, if you don't try to answer a question. You don't get points for that. You don't get points for showing up. This isn't participation based, really. So for doing this is why I want to clarify this, right? When I said I would call on you and ask you questions, I expect you to have the answer when I call on you, not say, I don't know. I'll give you a minute if you want to look it up in your notes. I'll give you a minute, right? But if other people start wanting to answer the question for you, I might have them answer the question for you. And if it was your job to answer the question, I might ask you to repeat their answer for me, and you can get your points that way. Okay, you can get your points that way. But what I found is that a majority of my class, the first class, couldn't answer the questions, and I had like 5 or 6 people that were carrying the weight for them. And I think that it came off pretty clear to them that that wasn't going to be acceptable. Right? When I say that I want participation, I mean for you to participate. We wrote our concept maps, right? Most of you guys did them pretty well. I mean, I made comments if I thought that you had some issue with it or whatever. I tried to be clear on that. If you didn't get any comments or just said great job, that meant good job. Okay. So so be aware of that. So if your concept maps are good, you can use those to help you answer your questions. Feel free to use your notes, whatever you need. You can ask your partner sitting next to you. This isn't meant to be like a quiz, but I do expect you to have some ability to have discussion about topics, right? You don't have to get the answers right, but if you're talking about the content, that's what I'm looking for, okay? The whole point of this is when you get on the test, none of the questions are going to be verbatim what you studied, right? Like, I think I made that clear, right? And so none of the questions are going to be verbatim what your flashcards are, what your concept map was. We have to start talking about that stuff so we can orient ourselves with that content better than just recalling it. Just recalling it, it's not enough, guys. It's just not. And it definitely won't be enough to get an A on my test. Like I guarantee you won't get an A on my test with recall. You have to apply it. You have to be able to put it into practice. And so I say it a lot in the beginning part of the semester because I want you guys to understand, like, I'm not joking about it. I'm not like saying it just because to make you guys do the work and then sit back and now you have an easy recall test and you overstudied for it. That's definitely not going to be the case. Tests are worth about 50% of my class, right? And historically, my classes get C's in my class, like as their final grade in my class, not on their test. I mean, on their final grade in my class. So that should put into perspective how poorly people do on the tests. So I'm trying to prepare you better for the test. And I'm absolutely not going to make my tests easier. And I do not curve anything in my class ever. And I never will. Do not ever bank on that. And I will fail you if I don't think you know the content well enough. I do it. How many people do I fail a semester? Maybe one each section, maybe. I mean, there's plenty of sections where I haven't failed anybody. Like I said, my summer class, all A's and B's, and they earned it. I'm telling you, you're getting the same exact course as they are. The same test, exactly the same test. I'll be honest with you, it's the same question. But are you going to go to one of the people that took my test and be able to get the test from them? No, because I don't open my tests. They were able to come to me after the test and I would go over the questions that they messed with them, but nobody ever gets the questions back or the answers back. I don't do that anymore because I don't trust people. I say it, but people just aren't trustworthy. I'm not saying it's you, but I am saying there are plenty of people in your classes that are not trustworthy. So, anyways, so that's why we do it. That's why I do the thing I do, because you are supposed to learn this and understand it and take it with you. Now that's true for any class that you're taking, especially your science classes, because like we said, raise your hands, guys. How many of you guys are going to some kind of healthcare after this? Right. And like we said, how many of your patients are going to behave exactly like the textbook told you? None, probably ever. So we've got to apply. So that's why I do this, because I take preparing you guys for that very seriously, because this is the time where you're going to get the opportunity to learn how to learn, because in the nursing program, they're going to teach you and you're going to get tested. They're not going to help you learn how to learn. They are not going to hold your hand in the nursing program. Anybody in here in the nursing program already? This is one class that I don't have somebody in the nursing program already, which is kind of not good for you guys. So if you don't know somebody that's already in the nursing program, if you're going into nursing, not the vet tech people, but if you're going into nursing and you don't have a contact in the nursing program, get one. Because I'm telling you, it is not good. It is cutthroat and the teachers are not there to hold your hand and they don't want to help you. They expect you to know how to learn. So I'm being serious about my job here. I am holding your hand. You guys think of it as just a lot of busy work. And I hear people talking about it. It's a lot of busy work, but if you're not treating this like a learning tool, then yeah, it feels like busy work. I understand the first week it probably felt a little bit like busy work because we want to just get it out of the way. If you don't learn how to use those as tools for your learning process, when you get to the nursing program, they're not going to have assignments that help you learn. They're going to have lectures and exams, and half of you guys are going to fail and not even make it through the program. I mean, the first semester, half of you guys will probably fail. This last round, do you guys, anybody know, everybody heard how many people made it through the first semester? Less than 60%. I mean, it was around 60%. It was like 57% or something like that of the students failed the first semester. of nursing. And that's the norm. So if you want to make it through your first semester, not have to retake and repay for those classes, because you don't get that money back. You don't get to retake it for free. I want to save you guys from that, but we got to start being serious about it. It might take you guys taking my first test to realize what I mean with this. That's okay. That's why the first test is 5% of your grade and not as much as any other test. All the other tests are worth more. That's why we have that set up that way. But we'll start to see what I'm expecting of you today. And that's okay if we're tripping a little bit today, right? My first class definitely did yesterday. They definitely tripped a little bit, but they made it through. I only had one person in my first class that just didn't answer a question. And I repeatedly tried to give, more opportunities for them to chime in, but they also didn't bring any notes or anything. They just sat there with nothing on their table. And it's just like, and if you guys are too nervous for me to call on you and ask you questions, you don't belong going into healthcare. Right though? Yeah. So I will be calling on you. If you don't know the answer, talk to me about the subject and you can save yourself, right? And if you really don't know the answer to that question, I'll come back to you with another question later. You get a couple opportunities for it. Okay, so we're going to go through it. Everybody ready? All right. So we had our come to Jesus meeting. That's all we did that. We're going to move on and get to business. All right, chapter 2. So if you have your chapter 2 notes or your concept map or anything, you can have that open and you can do this. Chapter 2. My Excel spreadsheet. And I will call on somebody for this one, right? So we'll go over this. Which type of chemical bonds have sharing electrons? You'll see what I mean here in a moment. This is all right, our first victim. So our answer is covalent. So covalent bonds are the ones where we actually share the electrons between the two atoms. So first victim here, Kennedy Armstrong. Are you here today? Kennedy's not here. So if you guys weren't here when I call on you, then I will come back to those people at the end. I'll cycle back. If she never comes in, then she doesn't get that credit for today, right? All right, so Mikey Kaiser. All right, is it Mikey? Okay. All right, so my question for you is, we know that covalent bonds are the ones that share electrons. What are ionic bonds?
A chemical bond in which electrons are transferred and not shared.
Right, so transferring or donating the electrons, if we actually are giving them. So my example, if you guys watched the lecture videos, and if you didn't, I'm not crying over it, okay? But if you get the time to watch the lecture video, all right? In the lecture videos, I use an example talking about me and my sister playing with Barbies. So when we have to share the Barbies, that's a covalent bond. When I'm hogging the Barbie, that's when electronegative atoms like oxygen hog the electrons. Now, we still have to share it. I have to let my sister play with it sometimes. She's the hydrogen atom, okay? But generally speaking, I'm spending more time with the Barbie, the electron. And so I would have what we call negative dipole moment, negative charge to general, okay? All right, because I'm hogging that electron. Electrons are negatively charged. I'm spending more time with it. I get a negative. Yes, negative polarity.
Just to clarify, oxygen is anionic, like ionization, the water. No, okay.
Sometimes. So sometimes. So here's the thing. So when we are H2O, right? You see it ever drawn like this, where we have a bond there. Like this is the bond, right? This is always when we draw that covalent. Always covalent. So if this is a covalent bond, like we said in this question here, that's sharing the electrons, right? But you guys have also learned that in water, we're not always just H2O. Sometimes in water, we're H plus and OH negative, right? When this happens, this is ionization. So that is ionization. So that can happen because oxygen is so greedy that instead of sharing, it sometimes just says, screw this, I'm off. I want this all the time. And so it does do that sometimes. And so water, when we talk about H2O, it kind of can go between these two. It can kind of go between. So yes, so that's a good question because there's gray area in chemistry, of course there is. So that makes it all just that much more complicated. So it's not wrong, but it's also not completely right either to say that we ionize. Sometimes it does, and sometimes it doesn't. Sometimes it's covalent bond. Sometimes we're sharing, right? Generally speaking, if we talk about something like HCl, we know HCl is going to be H pluses and Cl minuses, right? Like our acid HCl, hydrochloric acid, because that's an ionic bond that we have going between them. And so we expect that to readily dissociate. Some things are kind of in a gray area, which is like water. So I won't expect you really to understand about the gray area as much, but it is important for understanding this gray area when we talk about pH. Okay, so when we talk really briefly, and I know we're going to have questions about it, if I give a little primer on it, maybe when you get to you guys, if I call on you, then you'll have a little primer of the pH stuff reminding you about it, okay? I know you guys did a beautiful job drawing pH scales and telling me that pH runs from 0 to 14 and that 7 is neutral. Well, let's talk a little bit about why that is before we get into it, because this is something, and I tell you guys, we're going to use chemistry all the time. We're going to use chemistry all the time in this course, not the math, but the concepts, right? So the idea with chemistry, pH, when we have 0 to 14, can you guys see it okay if I draw over here? Because I'm gonna try to draw over here where you guys can see it. Okay. We have 0 to 14, and we know 7's in the middle, and we say it's towards 0 acid, right? I mean, I know, I'm just gonna make sure you guys know. And so 14 is a base or alkaline. But alkaline. Same thing. Base and alkaline is the same. Okay. And then 7 is neutral, we'll put an N there. Easy enough, most of us know this. All right. You might also remember that pH equals the negative log of the constant. This might mean nothing to you, but you probably recognize this. When we use these brackets, that means concentration, right? So the concentration of the hydrogen ions is here. That's one aspect. This is the most important aspect of pH. By far. Not OH negative. pH is a measure of the H pluses in there. Too many of you guys thinking, we get to 14, that means more OH negatives. You're not wrong, but what does more OH negatives mean? If you have more OH negatives, that means we're using up the H pluses. We're going to associate with the H pluses in the waters. And that uses up the H pluses, and that means less of it. So we'll get to that in a minute. I want to review this pH stuff first. This is the most important aspect of pH. Does anybody know what log means? You know it means logarithmic. I don't want to hear that. What does it actually mean in math? Really? There we go. It's base 10. It's that easy. Don't overthink it. It's based in tens. That is it. Do not overthink it beyond that. I know it's a fancy word. It's just based in tens. Okay. So we know this is based in tens. This is the hydrogen concentration. What's the negative represent? Does anybody know in this aspect?
Inverse.
Inverse. Thank you. Excellent. Inverse. All right. So what this really means is when you have a pH of 2, what you really have is 10 to the negative 2. What's 10 to the negative 2 in decimal points? 0. 10 to the 0 is 1. We know this, right? We should know that from math, right? 10 to the 0 is 1. 10 to the negative 1 is 0.1. Or you could think of it this way. If we think of 10 to the 2, if we're literally talking about 10 to the 2, what's 10 to the 2 mean? 10 2 times, right? Which is 100. Or how many zeros are after it? 2 zeros after it. Same thing with the negative. 10 to the negative 2 equals 1 over 100. It's inverting. That's what the negative means, just inverting it. All right, so if I say pH of 2 is 1 over 100, 2 zeros after it. That's what the 2 is. It's really, it is that. That's how many zeros are after it, right? Now if I say it's a pH of 10, I had to pick 10, right? Because of course I did, and I wanted to draw the 10 zeros. Equals 10 to the negative 10, which equals 1 over 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Which number is bigger? 1 over that or 1 over 100? 1 over 100 is the bigger number, right? pH of 2 versus pH of 10. When we're talking about hydrogen concentration, this is the hydrogen concentration. Literally. In math equation, that's what that's referring to. The hydrogen concentration, pH of 2 means 1/100 is your hydrogen concentration. pH of 10 means 1 over that. I'm not going to try. like 10 billion or something like that. I get it now. I'm going to try to figure it out. Oh, the pretty good is 10 billion. We tracking that? That makes sense. Yes.
Will we be using this in like?
You won't have to do the math or show any math work with it. I will never make you do a calculation beyond times 10 of something. Okay, so there will be times I'll ask you guys to do some multiplying or doubling. But I'll usually let you know beforehand. I'm not going to test you with pH and make you do pH calculations. I'm not going to make you do the calculations I say this, right? But you have to understand the theory behind it. Because I absolutely will give you a scenario like one that follows. You have a patient that comes into your ER, right? Normal, but I will tell you the test question. Normal body pH is anywhere from 7.35 to 7.45. Right? Your patient comes in and their blood pH is 7.2. Is that more hydrogen ions or less hydrogen ions? More hydrogen ions than what should be there. Okay, that's the type of question to expect. So it won't make you do the math, but you have to have perception of what's going on. Does that make sense? Yeah. Okay. So I won't make you calculate the exact concentration of the hydrogen ions, but having that perception of what's going on with the hydrogen ions and what pH actually means. Because so many of you guys get tripped up on like when I say the pH went down, what happened to the hydrogen ion concentration? And then you guys stop in your brains, you're like, oh, what do you mean? You weren't thinking of pH about what it really was the whole time. You're just thinking of it as a scale of 0 to 14. And unacceptable. You have to be thinking about what's actually going on in that scale and what those numbers mean, and keep that in perspective anytime we're talking about pH. This is absolutely key because if your patient's pH is down to 7, let's say, and it's supposed to be 7.35 to 7.45, and it's quite a narrow range, and if we're even below at all, we're going to be concerned. So if your patient's blood pH was 7, I would hope that they were in the ICU. That's very significant. Okay, so why is that dangerous to the patient though? Why is it dangerous for your pH, your blood pH to be going down? Anybody have any discussion on that? Yes.
How guess?
Yeah.
Does it have to do with like the proteins?
There you go. So what's your name? Gracie. Okay, Gracie, I ain't gonna call on you today because you talked about something and I need you to know. So, yes, so it does. If this will let me type, it's of course now my Bluetooth isn't coming. So yes, so it does. So what we're talking about here is essentially, why does it do that? Because I actually need the excuse me. Now I actually need the keyboard to work and it's not. It literally was working 2 minutes ago when I started this. Yeah. All right, sorry. So I can't do multiple things at once. I have ADHD, so we have to like get a thing done before I can talk about things. Yeah, so good. So H pluses, when we talk about proteins and how they fold, most of the time, and this is part of chapter 2 also, right? We learned about R groups in our proteins, okay? So with R groups, let's try to draw this on the board, but let me bring up the board just a little bit so we can see behind it. We know that your proteins, what are proteins made of? What are the building blocks of protein? Amino acids, beautiful. All right, an amino acid, normal amino acid, draw like an amino acid. You have your NH2 group at one end, just for the parts of it. I'm not gonna get super crazy with the stereochemistry or anything like that. We have an NH2 group, you have a central carbon, it always has a hydrogen, and we have that central carbon is attached to a carboxyl group, CO2. It's like this. If we draw, you guys know carboxyl groups, okay? It's a COH and a C double bonded to an O. That's a carboxyl group. And you know that this double bond can shift up to there, and that'll be the double bond. This will sometimes get an H. It's always kind of in between that. Chemistry stuff, love it, right? Anyways, you have your acid. This is your acid. It's a carboxylic acid, right? And this is your amino group. NH2 is amino. Amino acid. What's up at the C? You guys remember this? This is your R group. This is what makes your amino acid whatever it is. So if this is just a hydrogen, we call that alanine. You might have other ones. You don't have to memorize all of them. There's like alanine, isoleucine, leucine, methionine, cysteine, serine, glutamate, glutamine, aspartate, all of those guys. There's 20 of them. Did you have a question? Okay. Sorry, I saw your hand earlier. I didn't know who was questioning or not. So, right, so this is an amino acid. And that R group, some of them are polar. Some of them are actually literally charged, where they have an ionic bond going on somewhere with them. So remember, polar is different. We're holding on to our covalent bonds, but it's skewed. Somebody's hogging too much. Okay. Some of them are actually charged, where they actually donate the hydrogen typically. And some of them are non-polar. We learned in chemistry, non-polar molecules, how do they behave with water? Hydrophobic, right? They don't do well with water. Non-polar, because water is polar. We want to talk about it. The oxygen hogs the hydrogens, electrons, giving those hydrogens a positive polarity and the oxygen a negative polarity. And sometimes it even steals them. Right? So this is important. So if this R group, right? If we have our protein made up of a ton of amino acids, here's an amino acid, here's an amino acid, here's an amino acid, here's an amino acid, and so on and so forth for like 1,000 amino acids, then these R groups that they're attached to behave accordingly with each other. They might repel each other, or they might be attracted to each other, and stuff like that. And those things will depend on how much H pluses or OH minuses are around in that area. Now, we're expecting this to be a pH of 7.35 to 7.45. What we're talking about really is 10 to the negative 7.35 to 10 to the negative 7.45 as your concentration of your H pluses. That's really what the pH means. And so if your concentration is skewed outside of this, if we get to 7, like we said, 7 would be more H pluses than what we're expecting, right? So now you've got H pluses hanging around these guys that shouldn't be more. This should be at this ratio. We now have got more than we're supposed to. They start interfering with how those R groups interact next to each other. Or when we get that whole protein and how it folds, these folds are dictated usually by what kind of bonds? Hydrogen. And hydrogen bonds are literally just attractions between a hydrogen atom and something else that is usually oxygen because it's electronegative, something that has a negative. And so those are just attraction forces. So we have too many H pluses, the attraction forces don't happen the way that they're supposed to. So when somebody's blood pH is too low, they've got too many hydrogen ions in their blood, and now their proteins aren't going to function the way that they're supposed to in their body. We already talked about your genes in your body only code for proteins. You don't have genes that code for fat. You don't have genes that code for carbohydrates. You have genes that code for proteins. Not to mention, when we start talking about genetics, what does this look like? Let me draw. Sorry, right now wherever I can see it. What is this when I draw this? Okay, double helix. Right? So what's that representing? Your DNA, right? So that's easy enough. Most of us can recognize that. It's your DNA double helix. We'll put DH just to remember this is a double helix. So if we unwind that double helix, we've got two sides of a ladder, right? And those rungs of a ladder, you remember this stuff from chemistry, right? A binds with T and C binds with G. You know how I remember this? Is the C looks like the G. And the T has a crossbar to it like the A does. That's literally how I remember it. But how are the C's and the G's and the A's linked together on that bridge? Hydrogen. The only thing holding your DNA together. The only thing holding your DNA together is hydrogen bond. You get too many hydrogen bonds, what else can you screw up now? You get enough, too many H's, too many extra H's. If you get enough, now your DNA can't even hold itself because the H's are interfering. Instead of binding our G's and C's together, we're hanging out with H's that are just hanging out. You get enough acid in something. That's why if I put sulfuric acid and I pour it on my hand right here, the flesh just falls off basically because we've got so many H pluses that all the things that are holding my body together, what do you think is holding my body together? Hydrogen bonds. That's how acid degrades things. Now tell me that's not cool to actually think about it at that level. Literally 90% of what's holding the body, we have other bonds too, of course we do. Like a good portion, and I'm talking a massive, massive proportion of things that are holding your body together is just traction forces between a hydrogen and an oxygen. Wild. But that delicate balance depends on this. And so when you're bought a patient, blood pH is going off. That skews that. Yes.
I'm sorry.
No, you're fine.
Last night, I had a patient that was, I don't know if I'm saying this right, I wrote it down,
Neutropenia.
What exactly is that? 'Cause that's the first time I ever had a patient, but I know it has something to do with blood.
Right. So that's your neutrophils. And so when we're talking about neutropenia, we're talking about the level of the neutrophils in the blood. And so like neutrophilia would be neutrophils being too high. Neutropenia is the neutrophils are too low. And so your white blood cells, your number one white blood cells that are in your blood should be neutrophils. 55% of your white blood cells should be neutrophils. And so when we get above that, we suspect it's infection. The reason that they go up is infection. But did your patient happen to have an infection by any chance?
Yeah, so we had, I like down up everything.
Okay, so the fact that they have neutropenia meant that their white blood cells, their neutrophils, which are our first line defenders, their numbers were too low than what we expected them to be for somebody that had an infection, which is concerning, right? Because the only reason that you can go outside at all and not get sick is because of your immune system. And so if his immune system isn't acting the way that it's supposed to, he's not getting the protection from this immune system, or he or she, I'm just going to say he. And then he's not getting the protection that he needs from his immune system. So the whole reason that we can even be outside or in this room together or eat food and not get sick is because you have an immune system that protects you from those diseases. And if you don't know better, then you may have learned about something called SCID, which is severe combined immunodeficiency. And what that is you don't have any immune system at all. And so that usually leads to that boy in the bubble disease. Yes.
So people that have leukemia
Mhm.
are producing white blood cells
Mhm.
that can be found as being neutropenia.
They might. So it depends on how it's affecting them. So if you have like leukemia or something like that, usually what's happening is in the bone marrow, the precursor that led to those is somehow usually being affected. Okay, so leukocytes are all of your white blood cells. Lymphocytes are like your T and B cells, your more specific white blood cells. And so leukemia, it depends on what cell's being affected and at what stage in the development of those white blood cells. So that's a unit 3 thing. We'll talk about that stuff and you'll learn more about it in unit 3. But it's their precursor, what we call the hematopoietic precursor. You might have learned about this in some of your science classes, but literally all of your blood cells come from a hematopoietic precursor. And if that cancer was caused by something wrong with that precursor cell, then it could affect other white blood cells too, and not just the one that's growing out of control. It just depends on what stage we're talking about. Yeah.
That process starting in the bone marrow.
Yes, starts in the bone marrow.
Creation.
And even your red blood cells come from those. So any blood cell at all, white or red, comes from, and your platelets all come from that precursor cell. So there's something wrong with that precursor cell, all of those things can be affected. And sometimes cancers will start out as like one type of, we have stages in our stem cells, right? You have the hematopoietic one, where all of your blood cells come from it. Then you have other ones that are also still considered stem cells, but they're next level. These ones differentiate into white. These ones differentiate into red. These ones differentiate into your like neutrophil. These ones differentiate into T and B cells. And you can have something wrong at any of those stages. So depending on what stage something is wrong. All right. The people with SCID, which is severe combined immunodeficiency, they can't even make those any white blood cells at all. None at all. They don't have neutrophils, eosinophils, basophils, T cells, or B cells. So they literally can't make any immunity. So any exposure to any bacteria will kill them. Because any bacteria, even just environmental ones. So we say in the bubble, what that is, we try to keep them in a sterile environment. Even their food can't have bacteria on it. And they usually die at very young ages. We usually don't see people with that sort of severe immunodeficiency surviving into adulthood. It's very unlikely. You can imagine why. How do we keep all bacteria? And that's what I tell you guys too. It's like, how do you look at it as we're on today, where you can turn on this keyboard if you wanted to. Whenever we start talking about your immune system and all the things you're exposed to every day, literally, I take your immune system away from you now and you'll die today. You'll die today. I don't mean you'll get an infection. I mean, you'll die today. if I take all of your immune system away from you. And so even just going out into the environment, even just going out day-to-day stuff, whatever germs are on your steering wheel or in the air outside or in this room right now or on your pen or your iPhone or your clothes that you're wearing would kill you if you didn't have an immune system. So we're talking about exposure constantly to thousands and thousands of bacteria and viruses and fungi and protozoa constantly that your body is fighting off just for you to be able to live in here. And that's why we bring up the idea of vaccines. When people start saying, well, we vaccinate babies with like 10 things, that's got to be like an overload. Really? So, keep that in perspective. Every day, even as adults, where your immune system is weaker than a kid's immune system. Weaker, the older you get. You'll learn about this in anatomy. You guys ever learn about the thymus in anatomy? And how many of those people those bodies that you see in anatomy are old people and you might not even be able to see the thymus? It disappears the older you get. And it starts disappearing once you hit puberty pretty much. And it doesn't, that's where your T cells are trained. That's where your immune system is trained. I'm poking here because that's about where it is in your body. And most people don't even know anything about it. And that's where your T cells, that's where your immune system and your memory cells are going to get trained. It's where your captains for your immune response are going to get trained. So this is big picture stuff, right? So when we're talking about how that stuff applies with chemistry and everything like that, you're already getting the right idea. You're doing great. That's the type of thinking we want, okay? Okay, so we did this one. We'll move on to our next question. But this is the point of this, okay? Is to get us talking about this stuff. You guys are doing great. Really good questions. Hydrogen bonds. Tell me about hydrogen. Which of these choices best displays hydrogen bonds? Now be sure you read and understand the choices. This one got some people last time. Righty. So hydrogen bonds. I expected this because the other class did the same thing that you guys did. So hydrogen bonds are bonds that hold water molecules. I wasn't asking for the definition. So that's the thing. You guys are immediately looking for a definition. Stop that. We got to stop that because this is what the nursing school is going to give you. Every question will be like this. Don't look for this one looks like a definition. That's what you did. Now tell me. I'm going to call on a person. And you're going to tell me why. The yellow answer is wrong. Get ready. So is Kennedy here now? Kennedy? Still not here. Okay. Adolfo. Okay, Adolfo. Can you tell me why it's not the yellow one? You might have to think through it. We can talk through it too. So hydrogen bonds. If I told you it's the bonds that hold water molecules to each other, okay? But it isn't the bond that forms between hydrogen atoms in different molecules. So I'll help you out a little bit. Let's look at the wording of this statement. Between hydrogen atoms. Are hydrogen bonds forming between hydrogens? So there we are. So what are they forming between? There we go. So it's the wording of the statement. So it's not between hydrogen atoms of different molecules. It's between a hydrogen atom of one molecule and usually what other molecule? What kind of atom? Usually oxygen. There we go. Excellent. Good. Good thinking process. So you see how he didn't know it right away, but he was able to kind of talk through it with me. That's what we're looking for. That's okay. He didn't know right away. It's okay. And this is on me on purpose, picking the one that everybody else picked. Right? So we get that now, why that wasn't the right answer. But do hydrogen bonds form between water molecules? Yes. Between the hydrogen of one and the oxygen of another one. That's an attraction force there. You guys get that? Anybody have any questions about that before we move on?
Yes.
Yes.
So I get, because I answered incorrectly, because I was thinking that all the water molecules, I looked at that, but then yellow, because, so the way the question is in different molecules, so that means that it is a hydrogen atom. And not
One of them has to be a hydrogen.
any other kind of molecule.
Sure, but is it between hydrogen atoms? Between hydrogens, I didn't say between hydrogen atoms and other atoms, non-hydrogens and other molecules. I said between hydrogen atoms in different molecules. So you guys know that hydrogen bonds aren't between two hydrogens, right? About that, right? Because H's will have a plus charge. This plus attracts to plus, right? So that's what that's talking about there. So this is a wording thing, yes, but if we look at this, is this right? This is right. So yes, we're looking for the most right, and it is a wording thing, and it is a semantic thing, and I'm telling you, this is every question you're gonna get in nursing. I'm telling you. We have to be able to break down the choices. In nursing, it's worse than this, because every answer will be right. Every one of them, to some degree, but you have to find the most, I'm not kidding. Every question in nursing, every answer will be right to some degree. But what's the most right? And this one, the most right one, even though this is a semantics thing, this is semantics. This is reading and understanding. This is understanding the choice at a level where I'm not babying, right? I'm not going to word this always completely nicely because your patient isn't going to word things completely nicely because your situation isn't going to present them in a completely nice way. And you might not get all the information if you don't know how to predict things.
Because I struggled with hydrogen bonds in chemistry,
Sure.
so hydrogen bonds, do they only show, I mean, they show up in other kinds of things besides just water, but it's
Absolutely.
generally water?
So no,
Okay.
right, so when we looked at this, oh, I was gonna thought the thing was over here. That's what we're looking at. Okay, so we looked at this. That water?
No, I mean, I knew the, I mean.
Yeah, right.
But
So-
maybe there was water at play in the DNA or something.
No, so don't confuse yourself by thinking too much about that, right? So when we talk about hydrogen bonds, it's literally always gonna be, this is good, because you wanna break down, wanna understand it well enough that we don't get confused about it in other situations. So it always means there's something, whatever, there's something attached to an oxygen that's attached to a hydrogen somewhere. So where's the electron spending most of its time? Electron here is spending most of its time with the oxygen. So that oxygen has a negative charge. And what's the hydrogen that's covalently bonded to it have? Positive. Now this can get attracted. Something in, like this is where I say different molecules. So these aren't attracted to each other. They're covalently bonded. They're sharing electrons. That's not an attraction. They're actually sharing those electrons. But this hydrogen, if we had something onto a carbon, onto a hydrogen, would that hydrogen have a positive charge? Uh-oh. You guys don't know? Chart like this. Do any of those hydrogens have a positive charge? No, they're neutral. Because why? Because carbon is not electronegative. Carbon doesn't hog, but you should know oxygen hogs. We see it all the time, right? We see it. What's holding these guys together is oxygen hogging. When we talk about phosphates, y'all know what phosphates hogging. How do we know? Because P is bound to how many oxygens? 4 oxygens. All of these oxygens are hogging something. And so we'll talk about in class too, because you have to understand about, you guys remember this, right? You guys know what this is. If I draw this, and I draw this. You get it already, right? And these guys have 2 tails coming off them, but I'm not gonna draw every tail, okay? Because that's just, I don't have the patience for that. But you get it, right? This is, you guys know this. This is the phospholipid bilayer. What's the head got? They're phospholipids. What are the head? Not phosphorus. They are, how do you guys know this? Phosphate. And so each of these heads up here, what charge do they have? Negative. Each of those heads. If you guys don't know this, you did not learn chemistry. I don't know why you're here. I'm serious. This is the level you should know this before you come to my class. If you took chemistry and you dropped that, what business do you have coming to my class? I'm serious. The only class you had to take before this. And you thought you could drop it and just like forget it? Shame on you. Especially when you take physiology. You start learning how important your potassium concentration is, or your calcium, or your sodium. Those are all absolutely crucial to nerve signal responses. And number one thing, which we've talked about already, that we talk about being important for you staying alive, that can kill you so quickly. What's ATP? Energy, right? Energy, but what is it? Does anybody remember what it stands for?
Adenosine triphosphate.
Adenosine triphosphate. Adenosine triphosphate. Let's see, I found that. I'm writing this like an absolute idiot. You guys can call me in on that. All right. Adenosine triphosphate. So we're looking at the A. I don't even care what the A is right now. We'll get to that in chapter 6 in case you don't remember it. But here's our A, whatever. And it's linked to triphosphate. So we've got a PO4 and a PO4 and a PO4. Phosphate's always PO4. Now, if each of these is negative, and they're highly negative, and we're talking like 3 negative charges on these guys, right? Not just one. We're talking like negative. All right, south pole to south pole magnets, what do? Repel, right? They repel. Same thing with these, negative to negative. These guys are held together by what kind of bond? Covalent, right? They have to share electrons just to keep their valence happy, to keep those atoms happy. They have to share the electrons, right? Covalent bonds here. But it's like south pole to south pole magnets that are like bonded together. They hate it the whole time. They're repelling each other the whole time. And so when we cut off one of these phosphates from ATP down to ADP, and we remove one of those phosphates, usually moving it to something else. But when we do that, that's what releases energy. Because those constant pushing each other away, constantly living like that, they are constantly repelling one another here and here. And yeah, you can go down to ADP, which is adenosine diphosphate. And you can even go to AMP, which is adenosine monophosphate. And of course, you can just have adenosine. That's like what we have in our DNA. Right? Okay, you guys following why it's important to understand about those charges, right? We're important understanding now. If we can understand this in ATP, right? And you, again, like we said in class, you run out of ATP so quickly that if I suffocate you, you die within minutes. That's how much ATP your body needs constantly. Despite you being millions of cells that can all make ATP, if I suffocate you, in minutes you die. Because you run out of ATP, that's the only reason you die. So it's crazy, right? That's how dependent you are on that. All on this, too, on this repulsion and being able to cleave off one of these and release energy. Your whole body runs on that. That's how strong these negatives are. When we talk about your phospholipid bilayer down here, we have 100 phosphate groups. Now, are they going to repel each other? We can say yes, okay? Typically they would, but this isn't a force between negative and negatives repelling each other. This is a force between polar things. And what are the tails? Non-polar, there are, and then because we know they're non-polar, they are hydrophobic, right? Tails are, right? And that is going to be a more governing thing than negative and negative are, because they'd still rather be negative and negative near each other than have their non-polar tails interacting with water. That hates water. We know how bad our lipids hate water, because good luck mixing your oil in permanently with your water. Now, but can you get a negative to mix with a negative? You can probably get that to happen. But mixing the hydrophobic with water, that governs your lipid bilayer. And this will be key when we get into lab and we start understanding our stains. Right, so we'll come back to that for lab. But yes, so this can happen, and we'll have this set up, and then how these polar heads interact with water. Going back to your thing, this will interact with the H pluses of water. So yes, we have some water going on here. These oxygens, we have a negative polarity, okay? Negative polarity. So we can see how that would interact well with water, and this won't. And so this is important for understanding all of that. And so these hydrophobic tails, y'all sit there and tell me all the time about how these lipid tails are non-polar and they're hydrophobic. Why are they non-polar and hydrophobic? Because they made carbons and hydrogen. And carbons share equally. They're not like me and my sister. They're maybe like you and your sister. Maybe they share equally. And carbons always share equally. So now our H's don't get pluses to them because the carbons sharing equally. So that hydrogen stays happy with its little electron the way that it's supposed to be. No charge, no polarity. But oxygen hogs, carbon doesn't. Are we tracking a little bit how that organic chemistry stuff works? And so this is actually why organic molecules, you can't just have carbon. If I have carbon, CO2. Are these oxygens gonna hog a little bit? Probably, even from carbon. Right, but is this an organic molecule? Does anybody know, is carbon dioxide organic?
It's made of carbon and oxygen.
No, it has some hydrogen.
There you go, it's not an organic molecule. It has to have some hydrogen on the backbone of the carbon. So that's a trick, Okay, but don't fall for it, all right? All right, so we get there.
So it
True,
has to
true.
have Carbon and hydrogen.
The carbon has to have some hydrogen bound to it. At least one of the carbons do, for it to be organic. Okay? Okay. Is that helping at all with the hydrogen bonds can depend on some water, but they also can be between molecules that we hog the electron from. This could be bound to who knows what, a benzene ring, who knows what, but this, because the oxygen is hogging it, now this can associate with anything that has a negative charge to it, including maybe these phosphates. Maybe other things. There's a lot that can have an oxygen in it that has a negative charge to it, okay? It could be oxygen on other atoms as well. So when we start talking about carboxylic acid, for example, over here, and NH2 over here, NH2, this has a tendency to grab on another H and get a positive charge. And this has a tendency to drop this H and get a negative charge. Ionization. So we tend to have amino acids behaving like this. And so these can also associate with other things. But now you can see why this would be attracted to this on another amino acid. And so if we had that, if this O bound to another thing, that was another amino acid, just it's amino acid, okay? This is amino acid. This bond here between that amino acid and the next amino acid is called a peptide bond. Peptide bond. Between the amino group and the carboxylic acid up next. I know you guys learned this in chemistry, and I know it's review, but if they told you were gonna be good to forget it, they was lying. Because this is gonna come up all the time. Because all the time, I'm gonna be talking about this situation, our protein getting disrupted by our pH and all sorts of stuff. Unit 2 starts out pretty strong with a lot of this chemistry. A lot. And already in chapter 4, if we were actually paying attention, we learned a little bit about using some of our chemistry. We learned about the cell wall of our bacteria, which were made of, do you guys remember the word for it? What the cell wall of bacteria are made of? It's okay if you don't, because we're going to get to it in a little bit and you can look up those notes in a little bit, okay? We'll come back to the chemistry then. Before we move on, any questions about this? Is this helping a little bit? So we're taking this big picture, right? We're taking out the equations, not to give a crap about you doing some math. We're taking out the equations and just looking at the concepts. That's what I want you to be aware of. All right, let's move on. We did Adolfo, okay. Whew. But you guys are doing great. This is exactly what we're looking for, okay? Good, keep up the questions. All right, and what element is in all organic molecules? So this is in all of them. This should be the easy one, right? We talked about this just a second ago. All right, all organic molecules have to have carbon at least, but it's carbon that's linked to hydrogen, which we learned earlier, but it has to at least have carbon, right? At the very minimum, the carbon, and that carbon should be linked to a hydrogen, but you'll notice that hydrogen wasn't our choice on here. So we had several people choose oxygen. So our next person, our next volunteer, our next cheerful volunteer, Hannah Samuels. Are you here today, Hannah? Okay, Hannah. So oxygen, can organic molecules have oxygen in them? Yes, they can, right. So if we were to think of any examples, anybody think of an example of an organic molecule that has oxygen in it? Don't think too hard. So water doesn't have, it has oxygen, but it's not organic, right? But one we can think of might be sugars in general. You guys know all those H's and O's for the sugars, right? Definitely has oxygen. Nucleic acids, like we drew up here, those A, T's, and C's and G's, they're not just an A or a T or whatever, right? They are lots of carbons and hydrogens and oxygens associated with each other, right? Even ATP. We got carbons and hydrogens and oxygens all in here. So definitely we can have, so good answer. Good. All righty, excellent. Next question. You're doing great. Try to stay on point. We're already to 10 o'clock, believe it or not. You guys have good questions. All right, true or false? Water is a poor solvent. Not P-O-U-R, P-O-O-R. This one should be a pretty easy one. In our learning objectives, it literally kind of answers this. Don't worry, I'm not judging if you get it wrong, because we'll talk about it. But this one's false. Water is a great solvent, especially for polar molecules, right? And luckily, life is a lot dependent on polarity. So most of our life molecules are polar and they are very easily dissolved in water. All right, excellent. We'll move on. We won't have somebody, maybe we will, because these are easier questions to ask. But well, true false ones are harder to do. All right, what is water's pH? I know this one. So I'm gonna call on Tawny. Is Tawny Collins here? Okay, yeah. You'll be the person I call on next. We'll try to keep them relatively simple when I call on you. Yes. Did you get at least a couple questions in?
Yeah, well, Evans will know.
Okay, as long as you guys get one question in, I count you as absent, as present. Okay, so you can rejoin. Let's put it back up a little bit. If you need to rejoin,
I just updated one because I got it wrong.
so that you guys know when we do these Kahoot quizzes together, I'm not grading you based on the grade you get on your Kahoot, right? I'm just grading on participation for that, so don't worry about that when that happens. Yes. I just look to see that you participated for the Kahoot. So your grade consists of Kahoot participation and when I call on you participation. So if you have done a Kahoot, I don't care what your grade is, you'll get full credit for the Kahoot. But if I call on you, then I expect like actual participation, which you're doing great with so far, so no worries. I'll let you know if you're not. Okay. All right, so Tawny, if the pH is 3, that's lower than water. Is it more or less hydrogen ions?
And the pH is 3, and it's more.
Yes. Excellent. Right, so let's come back to that pH stuff that we've everybody clear on why that is? Yes. Right. I'll be happy to go over it again. All right. Next question. You guys are doing wonderful. What is the range? What range on the pH scale is an acid? Got all your choices here. Acids. I mean, it is written on the board somewhere in here. All right, so our acids are the 0 to 6.9 range. So is it Micherise? Micherise, okay. So I'm gonna ask you, which one of these, you can call it the color if you want to, okay? Which one of these would be a base? Red, yeah. Anything higher than so, right? So good job. All right, excellent. You guys are doing great. Next question in our chemistry chapter. Lipids, tell me about them. Here's your choices. Remember, there isn't all of the above choice here. On the exams, you won't have choice, you won't have questions where you have to choose multiple answers, but there might be an all of the above or none of the above. All right, so this one is all of the above, in case I didn't hint it enough. But that's okay. We know lipids can make up our cell membrane. We just talked about it over here. If we watch the chapter, then we should also know that they are used to make our hormones. So cholesterol and other sterols are usually a base to build off of to make things like testosterone and estradiol or whatever else, right? So can they be used to make energy for the cells? This is one that nobody picked. And I won't call on it, but this is a little bit of a complicated one, okay? But it can be fed into many of the steps of aerobic respiration, cellular respiration. Remember, glycolysis, Krebs, and electron transport chain. We learned about if you guys took GOB, you should have learned a little bit about that stuff. So we can actually break down some of our lipids to a molecule that can fit into one of those steps. And usually we'll break our lipid into acetyl-CoA. Like we'll break it into like a glycerol and then turn it into acetyl-CoA that can be fed into the Krebs cycle. If you don't remember that stuff, deep breath, okay? Chapter 10 reviews that stuff, okay? But do I expect you to know it? I'm not gonna ask you what, acetyl-CoA enters the Krebs cycle. What molecule does it become? Whatever. But you don't, I'm never gonna test you on that. I'm less concerned with each of those molecules and more concerned that you understand the point of them. We're not at chapter 10 yet. Deep breath. But we will get there, okay? We will get there. We're gonna put it in the context of actual living things. I'm less concerned about the other stuff, okay? All right, which of the following organic molecules is amphipathic? Talked about it already. Amphipathic. So which of these is amphipathic traditionally? One of these that the chapter definitely talked about. And Braley, I'm coming for you next, Braley. Is Braley here? Okay. All righty, so we got phospholipid. Most of us got that one, right? What does amphipathic mean? Braley?
It's both non-polar and polar.
Exactly. So it's got a polar aspect and a non-polar aspect. In this case, we can say a hydrophilic part and a hydrophobic part. So amphipathic always means, amphi always means both. Anytime you ever hear amphi, if we learned in our chapter 4 about our flagella, there was amphitrachus. Just remember that. Amphi, both ends. Amphi always means both. Like amphibians, live on both the land and the water. That's how I remember. I know it's silly, but these are the ways I remember things. Okay, so great job. Next, anybody have any questions about this one? Not two different charges, but hydrophobic and hydrophilic. That's what amphipathic means. Okay. A monomer of a nucleic acid. What are nucleic acids made of? So the parts that make up a nucleic acid. That's what a monomer is. Monomer are the parts that build a polymer. All righty, so nucleotides build our nucleic acids. There obviously is an example. I mean, this is an easy question, but I'm still going to ask it. Callista Brubeck, are you here today? I think that person hasn't joined any of our class stuff, so I just want to make sure they're not here all of a sudden. And Cora Holly. All right, you're Cora? All right, Cora. So why wasn't it DNA? So DNA was not the monomer of the nucleic acid. What is DNA in respect to this stuff? Want to make a guess? Okay, so DNA is a nucleic acid, right? And it's made up of nucleotides. So the A, T, C, G that we have over there, right? Those would be our nucleotide. It's made up of them. It is a nucleic acid. It's monomer. What it's made up of, its parts. Like we have proteins. You guys are really good with the proteins. You know, proteins are made of amino acids. Nucleic acids are made up of nucleotides. And chapter 6 will break that down more. So if you don't remember about nucleotides, chapter 6, that's a unit 2 thing. We'll go over that more. But remember, a nucleotide is some sort of 5-carbon sugar. Some sort of 5-carbon sugar, based usually around ribose. Some sort of phosphate, and whatever your nitrogenous base is. That makes it A, T, C, or G. Okay. That's a nucleotide. It's just made up of those. All right. We'll come back to you, Cora, no worries. All righty. Next question. We're almost done. This is our last question of this one. Alpha helices and beta pleated sheets are examples of which level of protein structure? If you know this one, Cora, feel free to chime in. You're okay. You're okay. If for some reason I call you guys and a situation happens like this for you, where you don't know the answer, you're not sure, you won't prepare for that today's class. Remember, the lowest, it happens. This is realistic, right? So if that happens to you, then number one, you can either remember one of the questions that I asked you and answer that question correctly. So tell me the question, answer it correctly in an email, just email me. Like I said, I'm not going to reach out to you for that, but if you do that, you can get your points back that way. Another way, what thing you can do, remember that your 2 lowest activities get dropped. If you mess one up, you're gold. Just do well on the other ones. Yeah? Keep that in mind. So there's not that much pressure there, okay? This is the secondary level. All right, Barry. Tell me, what is the primary structure of amino acid sequence? Or primary structure of protein. Can you define that one for me? Primary. You're fine. You're fine. I can just, like I said, address your notes. You can address the notes. The primary level of protein structure. So this one asks secondary. Yeah, I said it. I let it slip, and it really is that simple. Okay, literally the amino acid sequence. Whether it's isoleucine, leucine, alanine, methionine, cysteine, whatever, however you spell out that protein. with your amino acids. That's the primary level. Think of it as building your ribbon. Okay? When you curl your ribbon with your scissors, parts of it might be curled and parts of it might be straight. The curled parts are our alpha helices. The straightaway parts are the beta cleavage sheets. Okay? And they happen because of those R groups next to each other, repelling or attracting each other. All right? That's the secondary. Tertiary is tying your whole one ribbon, just one ribbon, tying it in a bow. But if you need multiple bows to make your package look nice, that's the quaternary. That makes sense? Okay. A lot of our proteins need multiple bows in order to function. So they often need that quaternary level. But if it's just one polypeptide and it's folded up in its tertiary structure, some of them function like that. Few, but some. All right. We tracking? All right, good. Because we'll learn more about proteins and enzymes in chapter 10. All right. Here's our little podium, and we're going to do our little activity. And I'm mostly going to walk you through the activity. But again, if you were somebody that either I missed you or you got a question wrong, if you can chime in with these answers, you can still get your points here too. Okay. So great job. We're going to get into our activity. I'm going to leave this up for now. You can let your phone sign out or stay on it. Either way is fine, because we'll come back to it afterwards and re-sign in for chapter 4. Because remember, we're going to do chapter 4 after this. I'm going to pull it up really quick just so that it's in the background. So it's just in the background. You can just leave it signed in. If you accidentally sign out, if you're fine. It will be fine. Okay, so we're coming back to chapter 2, activity. All right, so this is in that activity assignment. Now you guys, just to remind you guys don't have to submit anything for this. By being here and participating, you get the credit for this part, okay? All right. How are electrons distributed in a bond between highly electronegative atoms like oxygen and the hydrogen bonded to it? All right, so this is asking about that. Does anybody want to answer that? We kind of covered it. How are they distributed? So this isn't, don't read too far into it. This is asking about which one spends more time with the electrons. Oxygen does, right? Yes, so oxygen does. So we can say oxygen hogs the electrons in a covalent bond between oxygen and hydrogen. Okay. What is happening to hydrogen concentration in a solution where the pH goes down? Yes.
But the pH goes on.
Oh, goes up. There it is. There you caught yourself. All right. So what's your name? Shelby. Okay. So I won't call on you later because you answered a question out on your own. All right. All righty. Name the 4 major groups of biological macromolecules.
Carbohydrates, lipids, nucleic acids, and proteins.
Yes. Carbohydrates. You guys hear that? Carbohydrates, lipids, nucleic acids, proteins. Those are our 4 groups. What was your name?
Megan.
Okay. I know you're on here because I saw it just 2 seconds ago. Here it is. Okay. All right. So you got Megan. All right. Next question. Give one example of a hydrophobic molecule in biology that was discussed in the chapter. Anybody? Yes. Lipids. Okay, so that's a big group, right? So it's not wrong. So it's the right answer. So if we were to break down what a specific, give me one example of a specific type of lipid that's used in biology. Because we can say phospholipids. Okay, so that's easy enough, right? Another example might be cholesterol. Other examples might be triglycerides, right? So those are all kinds of lipids. And if we had one in chapter 4, there's a very unique one in chapter 4 that's more key than you probably realize when you're making your charts. You guys probably glanced over that mycolic acid stuff and was like, whoop, sure, acid, whatever, and moved on. But you have no idea how key and integral it's going to end up being. So in chapter 4, we talk about mycolic acid. And mycolic acid is a special kind of wax, which is another one in our microbes. That's a very important one. And we'll talk about it in chapter 4. We'll get to it, okay? But that works. All right. Question 5. Given what we have learned about proteins and their structures in this chapter, why might a decrease in pH, all right, negatively impact a protein's ability to function? Anybody want to try it? Yes, Tom. Denature them. And so how does that happen? So anybody want to try to go into a little bit further explanation? This is right. And I know she probably has the idea, but anybody else want to try to elaborate on that? Why does it denature the proteins? So I'm just looking for you to explain. So what, so what did you say? Sorry, what did you say? Hydrogen bonds. Okay, yeah, sorry, I just couldn't hear you the first time from my hearing. So yes, hydrogen bonds. Those hydrogen bonds that cause our groups to associate with each other and cause that folding get disrupted by low pH, decrease in pH, increase in H pluses. So these H pluses are interfering with our ability of our you see how this is, we need to start thinking about things in that respect, right? We talked about this, I know we got it, but putting that into words, actually having to verbalize it, you guys have to think through it and make sense of it in your brains. So that's the whole point of this class and why we have it this way. All right, so what was your name? Okay, right, okay. So I marked you down earlier, okay, good. Just want to make sure. All right, so we'll come to chapter 4 in a minute. We're going to do the chapter 4 Kahoot. So just double check that you're logged in. Or log back in, whatever. We're going to do chapter 4 really quick. Say really quick, but we're here for another 50 minutes or whatever, right? 40 minutes. All right, you guys are doing great. You're hanging in there. Take a breath. And we're going to get moving on. We still got a few people to call on. All right. While you guys are signing in, I got a text message from my brother-in-law. He's going through chemotherapy for testicular cancer. And he's going through his last cycle of chemo. He started his last cycle of chemo today, so we're very excited. I know. He's doing well. His blood work is great. By the way, if you don't know much about cancers and like the difficulties with different cancers, if you needed to have a cancer, testicular is the one to have. It's like one of the most treatable. It's like 99% treatable with chemotherapy alone.
This is called relate.
Huh?
Right, this is called relate.
Yeah, so it's like, so we will, so I'll say this is like, if it's caught early enough, but even if you start having symptoms of it, typically, depending on what stage you're at, right? Even if it has metastasized, it's usually treatable with therapy. But if you're at stage 4, of course, then, right. But usually you're going to catch it earlier than that. Most people have symptoms of some kind before they get to stage 4, like this.
So brother had his prostate removed, and then he came back to his test, and they came back to be really high.
Oh, yeah, yeah. Yeah, prostate's a little different. So prostate is definitely not as safe to have as testicular. So the testicular, if it's originating from testicular cancer, that's a more curable cancer. If you have to have a cancer, I'm just saying. And just to be clear, if you guys are thinking of like, okay, if you have to have a cancer, 1 in 2 people will get cancer in their lifetime. I know, that's a real statistic. And it's because we're living so long. So yeah, the longer you live, the more likely you are to have cancer, obviously. I'm going to talk about increase of melanoma right now, just a little mini break before we get going in this. Melanoma is increasing right now, and people are saying like that's because, there's big arguments right now about how sunscreen doesn't actually work and how it's just a gimmick by consumerism. But that's not true. It is not true. Okay, the reason melanoma is going up is because the millennials like me didn't wear sunscreen when they were younger, and now they're getting melanoma. Not because Gen Z is using sunscreen. It's not Gen Z that's getting the melanoma. It's the millennials and Gen X that's getting melanoma. Okay, or the boomers. Because we didn't use sunscreen, I can assure you. Elder millennials did not use sunscreen. We poo-pooed on that. And y'all stick with your sunscreen. You don't want to get skin cancer because melanoma is not a cancer that you want to get. It is a deadly one. And go to your doctors and get checked. And wear sunscreen. All right. Talking about with my students that were talking about like being older and like how weird it feels when you start realizing how old you are and you get to that phase in your life and you realize that you're older than a lot of people and they haven't seen things that you've seen, right? Okay. So I was talking about how one of my earliest memories, that is one of my earliest ones. Okay. So those of you who are a little older than me, don't laugh too hard at it. Okay? But one of my earliest memories that I have, and I mean when I was little, was Chernobyl. I remember Chernobyl. I am that old. I vaguely remember it. I was little when it happened, but I remember seeing it on the TV and my parents being scared. I didn't really have context for it at the time. But I look back on it now and I know, you know, I have better context now. Most of you guys weren't even born then. Some of you were. I'm not gonna.
What year?
That was 86.
Some of us-
Some of us remember, yeah. Some of us. If we were to do that, we could put that into perspective, but it's gonna be more of us probably in here that were alive for 9/11. But I definitely remember 9/11 quite vividly. Yeah. Anyways, yeah, we won't do that though. All right, let's get it going with our thing. Yeah, most people are surprised to hear that I remember Chernobyl though. That was,
The major tornado.
yes, I remember that. That was 2000, wasn't it? I think it was.
Yeah. I think so.
I don't know,
99
or
or
99 or
2000,
something like that. 99, yeah,
3 mile wide one.
yeah. It started in western Oklahoma and stayed on. I mean, I'll be honest, I lived in Tulsa back then, so I didn't care as much. I'm just saying, I had a different perspective on the tornado stuff there. Tornadoes rarely make it to Tulsa. Anyways, so it really is, like, if you guys haven't, if you've lived in Oklahoma City most of your life, I'm telling you, Tulsa, it rarely makes it to Tulsa. We'll take shelter a couple times, but there's not the same. I've lived in Oklahoma City most of my life, but the first part of my life was all in Tulsa, and it's different. Tornado season is different there, and it's not that far away. So I'm telling you, it's not as scary there, because it rarely is an issue in Tulsa. I say that, but my parents' house was hit by a tornado in Tulsa, so just it's not 100% safer, but this does put a different perspective on it. All right, so our question was, since bacteria don't have a nucleus, they can't contain DNA. All right, this one was false. So I'm gonna call on somebody. Mariah, are you here today? Mariah Brown? No, Mariah, okay. All right, so our next person, Victoria Vincent, are you here today? All right, Victoria. Do you want to try on this one? Bacteria don't have a nucleus. They can't contain DNA. This was false. So they don't have a nucleus. Can you tell me what a nucleus is? It's okay, don't overthink it. So when we talked about eukaryotes and eukaryotes, we talked, yes, you can look at your notes. So I'll give you a minute. So we talked about eukaryotes and eukaryotes. We talked about the difference in the nucleus, right? And we talked a little bit about what a nucleus was. We'll give her a minute. Is she working? Is she cooking? Let her cook. Let her cook. What is a nucleus? So we have here, bacteria they don't have a nucleus. That part is true. So we also, you're all right, take your time. So I'll go on to the second part of it while you're looking at your nucleus part, okay? Second part is the DNA. If you're a cell, any cell, whether you're eukaryote or eukaryote, there's only two types of cells, eukaryotes and eukaryotes. But they're both cells. All cells have double-stranded DNA, just like that. All cells have that. All right. Have you figured out the nucleus yet?
Okay, so it's not this thing.
So it is central. We'll think of it as the control center of the cell. And when a cell has a nucleus, there's one defining characteristic about it. So if you can figure that one out. So membrane bound. Membrane bound. So it's all right. We'll come back. We'll give you another chance. Okay. It's all right. So you're exactly. Because it's not like you're not participating, right? So definitely don't worry. We'll come back to you. Okay. Or if you know one, let I ask later. Just chime in. Right. So bacteria, they don't have a nucleus, but they do have DNA. So all cells do. So where their DNA is just hanging out in the cytoplasm. Okay. It's just not bound by a membrane. We call it, there's a region of the cell where it hangs out, called the nucleoid region, but it's just a region. It's not an actual defined structure. All right, good. We're getting there. We're learning a little bit about bacteria on the way. Come back. See if this next question is any better. Peptidoglycan layer in gram positives compared to gram negatives. This is our bread and butter. For sure, if you didn't get this on your, you probably made it on your concept map, but if it didn't stick, it will by the end of the semester, I can guarantee you. We're gonna talk about it all the time. All the time. Peptidoglycan layer. So that is, but I didn't want to give it away, right?
I'm sorry.
All right, so I'm gonna ask, we'll give you a chance here, Victoria. What does peptidoglycan do in bacterial cells? What's it make up? What part of the cell? So we know it's a layer. Peptidoglycan. And then we'll have one more question for somebody else. So if that layer is thicker in gram positives, our other person, while she cooks, our other person is Esther. Is Esther? Yes, do you have a question?
No, I'm
Oh, you're gonna ask me? Real quick, we'll ask, is Esther here today? Okay, Esther. All right. Gram negatives. All right. If the peptidoglycan layer is thicker in gram positives, you know the gram negatives are thinner. Peptidoglycan, that's good, right? What else do gram negatives have? Another defining feature about the gram negatives. We got some people that have answers probably. We're gonna give Victoria and Esther a second. So, what did you find out about peptidoglycan? What's it do?
It's like the armor of bacteria.
That's close. Okay, so it's the cell wall. So I'll give you that. Okay. Yes. So it is the cell wall. We have a thick peptidoglycan wall around our gram positives, and a thin one around the gram negatives. And the gram negatives also have. Okay, so both of them have the inner cytoplasmic membrane. I'm going to give that to you. I'm going to give that to you because they do have that. Both of them have that. What else do gram negatives have? Right, which is part of, yes, the outer membrane. Okay, let's draw it. Because I know this is a lot all at once. We're going to draw it so that you guys can see. All right, here is a cell and here is a cell. They are both cells. One of them is gram positive. We'll put a G plus. And one of them is gram negative. We'll put a G negative. The writing is going to be terrible, and this is why I don't write on this. Okay. But anyways, all righty. So we're going to try to go. I want it thick, real thick, and I want a different color. Do purple. All right. Our gram positives have a thick, this is peptidoglycan in purple, okay? Gram positives have a thick peptidoglycan layer around them, whereas gram negatives have a very thin one, real thin. But gram negatives also have an outer membrane. This, and we'll come back to this color because it's about the same. Outer membrane. It's also a lipid bilayer, both of them are. Okay. So see how we have different outsides on both of these. Both of them have an inner cytoplasmic membrane. That inner membrane, the cytoplasmic membrane, holds the cytoplasm in there. Just holds the goo and everything in there. They both have that. The wall is thick on gram positives. The gram negatives have a thin wall and an outer membrane to make up for that. So that wall gives more stability and rigidity. With that outer membrane, they're less rigid, but they can survive different conditions. Okay, so gram positives, more resistant to drying, more resistant to salty things because that rigidity helps them hold their shape. But the gram negatives live better in your gut and in acid, in like areas where there's going to be acid or areas where there might be bile or other things like that. So they're better off in your gut than the gram positives are. And this is, like I said, our bread and butter in bacteriology, which most of this class talks about bacteria. Talk plenty about viruses and protozoa and things too, but definitely a lot about bacteria because they're just a lot different than our cells. Okay, so that aspect of whether you have a thick wall or a thin wall with an outer membrane defines whether you're gram positive or gram negative. All right?
Which one's harder to treat?
You can't really compare them like that. So it just depends on the situation, where the infection is, what the actual microbe is outside of that. So I'll say this. Thick wall, we talked about this last time, right? Thick wall like that, we have something like penicillin, the cillins that inhibit cell wall synthesis. They need the thick wall. It's thick for a reason. So if we give them penicillin that inhibits cell wall synthesis, then that'll treat gram positives, and then your gut bacteria will be happy. They don't get killed by that as easily. So you don't kill off as many as your gut bacteria, so it saves you. In fact, that's one of our best treatments that we have against bacteria is those ones that attack the cell wall. They're not as effective for the gram positives. If I push enough of it, we can eventually treat some gram negatives too. Okay. So we'll use other antibiotics usually to treat gram negative infections. So you might have noticed this. If you get a UTI, 80 to 90% of UTIs are caused by the E. coli from your own butt. E. coli. And E. coli is gram negative. So we might not reach for a penicillin for E. coli infections. So the outer layer in a gram negative is just the outer layer. So we'll come back to that in just a second, Maria. I'll talk on that in just a second because we're going to talk about that in a moment, okay? But I want to stay real quick on this other thing, okay? We might not reach for penicillins to treat a urinary tract infection. In fact, what do we treat urinary tract infections for? Does anybody in here know? I know somebody in here knows. Bactrim. Okay, Bactrim is a medicine called nitrofurantoin. But it ain't a penicillin. We'll just point that out. All right, so it works in a different way. I won't make you know how it works right now, okay? But nitrofurantoin is designed to basically accumulate in, like it doesn't get removed from your urine. It accumulates in your urine, so it's concentrated in your bladder. And so it doesn't really circulate in your bloodstream. It gets concentrated in your bladder where we need it. And then in that area, it works on the cells. You don't need to know how right now. We're not gonna get into it. We'll talk about it later, okay? But it doesn't treat gram positives, it treats gram negatives. So Maria asked online what that outer membrane is called. It's just called an outer membrane. It's a lipid bilayer. But in that lipid bilayer, like we talked about just a second ago, remember how somebody brought up endotoxin? Endotoxin, endotoxin is also known as what Maria was just bringing up, LPS. Now I understand in your book, it talks about how endotoxin is part of your LPS. It's the lipid A part of the lipopolysaccharide, but I'm not going to test you on that. I just need you to know that they're relatively equivalent. Okay, so if we talk about lipopolysaccharide, we're also talking about endotoxin. Okay, and I will often talk about them both at the same time. And probably by the end of this course, that'll be another thing that you won't question anymore. So I talk about it a lot. Okay. Gram negatives in their outer membrane, they have lipopolysaccharide, LPS, which is also known as endotoxin. And endotoxin causes your body, the second it sees endotoxin, your body can see some things. I usually wear a lot of bracelets, and of course today I didn't wear any bracelets. Let me see if I have anything that I have multiple of. We'll say this. Do these look different from each other? Do they look exactly the same? Whatever. Okay, so I usually wear multiple bracelets. Do these erasers. Yeah, no erasers. Right, your body can look at these erasers. I'm going to bring it over here for the camera, people online, okay? Erasers. Your body, your innate immune system can tell me these are erasers. Your adaptive immune system, also known as your selective or acquired immunity, your B and your T cells. Your B and your T cells and the antibodies that your B cells make can tell me which eraser is which. You see the difference? So endotoxin is something that your innate immune system, like your neutrophils, use to say, this is a bad guy. It is a marker that you help you identify that these are erasers. Erasers don't belong here. Get them out. Endotoxin is a marker to let your body know this is something that is not self. We should think about getting rid of it. Then your B and T cells get activated because we're responding to, hey, there's erasers here. And your B and T cells target this eraser and this eraser and this eraser specifically with antibodies that see this eraser, antibodies that see this eraser, antibodies that see the third eraser. So we'll talk about that in Unit 3, but endotoxin is part of that response, okay? So your body can see endotoxin and say, bam, you're not self. Now, when I say that, all gram negatives have endotoxin, including the gram negatives in your gut. So we had to train our body. We don't react that way to the gut. The gut is kind of a unique site. And so we had to, your gut bacteria actually helped train your body on when and where to react to these things. But if you get endotoxin in your bloodstream, your body is not having it. Endotoxin in your gut, though, we're okay. just stay there. So your body knows better. And it got trained to deal with that. So that's endotoxin. I'm going to leave this up on the screen for now. It's kind of going to be weird in a weird place, but that's okay. It's there to remind us, okay? The gram-negative outer membrane contains Is it kick you out or something? It's all right, you can rejoin, and if you can't keep going with it, that's okay, you can just pay attention, as long as you got a couple answers questions. You know what I'm saying?
If you putting it access, like, what is it just like?
Yes, yes. So if you go to the lecture activity exercise, which you'll have to like look at it for it in your assignments or on the modules, because it's not going to appear in your to-do list, okay? But if you go to it in your modules or at the very bottom of your assignments list, you can click on that assignment and it has the Kahoot link in there. Just use a different username if you do it. Okay. All right. Endotoxins. So yes, we know this was endotoxins is written over here. So I'm going to ask Emma a question. Emma here. Oh, sorry, I have no idea. Emma Wilson. Okay, so you're Emma Wilson. All right. So endotoxins are found in the gram-negative outer membrane. What are endotoxins also known as? I know I have LPS, but do you remember the fancy word for it? Okay, that's okay. That's okay. So let's see. Lipoteichoic. Do you know where that's found? I'll give you a hint. It's not gram negatives. It's gram positives. Okay, so that's enough. We get there. We're getting there. We're making the connections. All right. It's a gram positive. So in that wall, the gram positive wall, like we have drawn over here, it's so thick. It's basically like levels and levels of scaffolding. We'll try to draw it again because like, honestly, it's worth me drawing, but it takes me a minute. Okay. So pretend these are sugar molecules. Okay. I'm not going to draw the whole hexagon, but this is hexagons. Okay. There's sugars and each sugar is different. It's NAM and NAG. Repeat, I just did really bad drawing. Okay, whatever. These are supposed to be chains of sugars linked together next to each other in a big long chain. And here's another chain, whatever. Here's a sugar hexagon, for sure that is a hexagon sugar. NAG and NAM alternate. I'm not going to test you on that on the test. It is in your textbook for those of you that care. NAG is N-acetyl-glutamate and NAM is N-acetylmuramic acid. I'm not going to test you on it, but those are the repeating sugars in peptidoglycan. Again, peptidoglycan actually tells you what it is. Glycan, what kind of molecule does that make you guys think of? Sugars. Yeah, like glucose sugars. And you're right, that is. So it's repeating sugars. And what does peptido make you think of? Peptides. All right, and so what are peptides? They are amino acids that are linked together. So we have peptide bridges. A few amino acids, a handful, like 7 amino acids, just linked together to create a bridge between our sugar chains. So this creates a scaffolding pretty quickly. That's what peptidoglycan is. It's really not that complicated, right? It's just sugar chain linked by peptide bridges. That's it. But in our gram positives, we have layer on layer on layer on layer of those things. And it gets so thick and unmanageable, we have these special molecules called teichoic and lipoteichoic acid just to help manage it all. So we don't have that. We don't need it in our gram negatives because they've only got like one layer of peptidoglycan. This is actually the principle on which the gram stain works, which we'll get to in a moment. We'll see if we get there. All right, so we got that. I'm going to leave that up there, like I said. All right. Don't tell me now, keyboard. All right, what is acid-fast staining? This is actually an image of an acid-fast stain. Acid-fast staining. What was earlier? Let's see if we can remember. All right. Staining to determine the presence of mycolic acid. So our next person is Jessica Morales. Jessica, all right. Jessica, mycolic acid. Do you remember what kind of molecule it is? Like in our biological macromolecules. I'll also ask you another thing. Do you either that or do you know what species of bacteria have mycolic acid? Either one of those. So we'll let you cook. We'll let you cook. So acid-fast staining is a differential staining procedure that we use to basically identify mycolic acid. We know that on here, right? All right, so mycolic acid is a special molecule that's only found on 2 genera of all of our bacteria. So one of them is nocardia. Do you know what the other one is?
Would it be full bacteria?
Yes, perfect. Okay, excellent. So mycobacterium, I will save you from having to be called on later on, okay? So yes, so mycobacterium. So this is a thing that I know a lot of people were getting a little confused on already, and I want to be sure that we're clarifying it before things get too tricky, right? Mycobacterium versus mycoplasma. Don't focus on the myco part. I know we want to lean into myco and think automatically that means mycolic acid. Don't. First of all, I'm never going to test you on nocardia. Let that one slip away into the wild blue yonder. All right. But I will test you on mycobacterium. Anybody know what kind of disease? Let's see if Brandon knows. Yeah. What kind of, yes, that's a good question, right? So what kind of disease? Name one disease caused by a mycobacterium. Leprosy is one. Does anybody know another one? Tuberculosis. I'm sure we had a lot of hands go up for that one, right? It's just tuberculosis is another big one. So Mycobacterium tuberculosis and Mycobacterium leprae cause those respective diseases, okay? So it's pretty easy one. So Mycobacterium, they're always called Mycobacterium blah, blah, blah. So if it is called Mycobacterium blah, 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 they always have mycolic acid. This is how we're going to define this, okay? I'm going to try, and you're going to try to follow this with me. Mycobacterium, I want you to focus on the bacteria part, the second part. It's bacteria. That doesn't tell us a lot, right? It's bacteria, cool story. Then you come to the myco. It's bacteria with myco, which is mycolic acid. Mycolic acid is a certain kind of wax that is found on Mycobacterium, which are gram positives. They have a thick wall, and in their wall, they have this wax on it. It makes them resistant and hardy and things like that. Okay, that's what we need to know about it for now. We use a special staining procedure to stain it because it's a special wax, it's a lipid. We use lipid soluble stain called carbonfusion, which we'll do that in lab 4, which is next week, okay, next Thursday. So we don't have class on Tuesday. You guys know that, right? Just gonna remind you, no class on Tuesday. We have class this Thursday.
So Monday and Tuesday?
Yes, because I want to keep you guys on the same schedule as my other class. Okay, because otherwise they get an extra day off. Did you guys notice that? They get an extra day off compared to your Monday, Wednesday classes versus Tuesday, Thursday. Yeah, so you guys get that day off because it keeps you on the same schedule. Okay.
Sweet.
Yeah, I know, I'm not complaining either.
You know, homework done.
Right, and you guys know you don't have any concept maps due this week, right? Okay, so I know. Take a breath. And then when you pick back up with them again, it should be 2. Right? Anyways, moving on. Mycolic acid, that's a special wax, and we use a special stain that's lipid soluble to stain the wax. Other normal cells won't stain with it. They've got this and just regular peptidoglycan going on. All right. Tuberculosis, Mycobacterium tuberculosis. Okay, that was what I was leading into, the mycoplasmas. So myco, if you hear a myco name of something, Mycobacterium, focus on bacterium. Okay, bacteria with mycolic acid, right? If you hear mycoplasma, focus on the plasma. The plasma will tell you something. I'm gonna tell you what, if I'm carrying a cup, if this is a cup of plasma, so it's an open cup, pretend with me, right? It's an open cup and I'm walking around with my open cup of plasma. You guys know what plasma is, right? The liquid portion of blood. So that's literally what I'm talking about here, right? So I'm walking around with a liquid portion of blood and I drop it. Is that liquid gonna hold its shape? Neither are the mycoplasmas. Because the mycoplasmas, they don't have anybody know? Let's see if I can call one of these people. Kristen, Morgan, Lily, Caitlin, or Anastasia. Any of you guys know? Yes? They don't have a cell wall. What's your name? Okay, Kristen. Thank you. Mycoplasmas don't have a cell wall at all. They're gram negatives, but instead of that little purple layer in there, they just got nothing. And so they don't hold their shape. They don't have any cell wall to help them hold their shape. So they don't have a real shape. We call that pleomorphic. Pleomorphic. Morph is shape, pleo is many. Okay. We tracking a little bit. So that's how I remember the difference between mycobacterium and mycoplasma. Anybody know what mycoplasmas are associated with? What kind of diseases? So I'll give you a hint, it's pneumonia. And it's called Mycoplasma pneumoniae, that's the name of the organism, okay? But it causes a certain kind of pneumonia called walking pneumonia. So what we call atypical pneumonia. So if you have pneumonia, which is fluid in your lungs caused by an infection, and you're able to otherwise function, maybe you'll feel like crap, but you can otherwise function. You're not hospitalized. You're not cyanotic to the point that you have to be on a respirator. Okay, you're not being treated in a hospital, but you can function otherwise. It's usually caused by Mycoplasma pneumoniae. So it is medically relevant. Okay. All right, good job, because I think that's a question coming up. True or false, mycoplasma is acid fast with a thick waxy cell wall. You guys paying attention? Mycoplasmas, do they have a waxy cell wall? Are they acid fast? So that acid fast stain that we talked about with our mycobacterium, not the mycoplasma. This is why we want to parse this out, okay? So the mycobacteria have mycolic acid wax on their thick cell walls. Mycoplasmas do not have a cell wall. That's why they are pleomorphic. They don't hold their shape, just like the cup of plasma. Okay, we're tracking? All right, Morgan, are you here today? Morgan Maxwell. All right, can you explain that back to me? That's what we're going to do here. Why? Why is this false? Exactly. Okay, good. So let's not get those mixed up. Let's just make a place in your mind where you divide those between each other. All right, question 6. What are bacilli? So this is a good question. So we're going to talk about bacilli here. I'm going to call on Lily here in just a little bit to tell me about another shape. Bacilli. The bacilli is a shape of cell. These are rod-shaped cells are called bacilli. And we're going to talk about it all the time. Again, another thing that we're going to use all the time. We're going to do it in the lab today too. But Lily, are you here today? Okay, Lily, I'm going to have you tell me, what do we call round-shaped bacteria? There we go, coccus. Coccus is singular and cocci is plural. So if you have a lot of them on there, we'd say that's cocci. Make sense? A bacillus is, all right, excellent. If an organism is named bacillus something, that tells you that what its shape is. But it's also its name, which is a different name. And this relates to another thing which we learned about in that chapter. Oh, sorry, I skipped over the thing. What does peritrichus mean? Caitlin King, I'm going to be calling on you about flagellar arrangement. Yeah, pull your notes up. Get ready. So peritrichus. All righty, these are flagella distributed over the entire cell. But I'm going to ask Caitlin. Caitlin, where are you? All right, Caitlin, can you tell me the word for when we have a flagella at only one pole? One flagella at one pole. One flagella. We'll let you cook for a second. There's 4 different arrangements that we can have a flagella. If you have a flagella, or you might not have one, and you don't fall into one of those, okay? If you are a motile bacteria, motile means you're able to move around. Bacteria use flagella to do that. So if you're motile, you have a flagella, you have at least one. Peritrichus means you have them all over your surface, all over. And the more you have, the more you can control your direction. All right, so flagella move by rotating. They don't whip. I know you want to think they do, but they don't. They only rotate one way or another way. And when they switch is when we cause the tumbles, the runs and tumbles. We'll be rotating one way, shuttling along, and then we switch the direction. That causes a tumble, and they can control their runs and their tumbles, and that's how they control their direction. It's really non-specific. But the more that you're covered in flagella, the more you can have some on one side of your body rotating and maybe not on the other. You can kind of control how you're moving a little better. So that's how bacteria move. We'll talk about how the eukaryotes move in a different chapter, but they can whip, they can actually control them. Okay. But if you are a bacteria, the only thing that you have to move yourself around with is flagella. They do not move like amoebas, where they kind of like scoop part of their cell apart. They don't do that. That's called pseudopodia. They don't do that. They don't have cilia, which are little hairs. They don't have that. The only thing they have for moving is flagella. If you don't have flagella, you don't move. Okay. We have 4 arrangements of flagella. Peritrichus, you're covered in them. All right. Lophotrichus, you have a tuft. Amphitrichus, each end. And Caitlin, can you tell me about what one flagella is called? One flagella at one end. I'll give you a hint. It ends with trichus. So if you have one of something, what kind of prefix might you use on it? Monotrichus. Okay, you're good. So we talked about some of the other ones, so I'll ask you just so you can get your points for today. What does lophotrichus mean? Which one is lophotrichus? All right. All right, it's the tuft. The tuft. All right, so you know what your question is going to be, what to email me with, right? All right. If you want, I mean, you could skip it too, it's fine. Remember, the lowest ones will get dropped. All right, so next question, let's move on. We got peritrichus, lophotrichus, monotrichus, and amphitrichus. Bacterial capsules. Tell me about the capsules. The capsule is not the wall. The capsule is not the membrane. Capsule is something else. Tell me about it. Which of these applies to the capsule? And Anastasia, I'm going to call on. It's okay. It's a good room, I think, more than anything. As long as you got one in, I got you covered. All right. So, bacterial capsule is a sticky coating. Usually, the capsule falls under a category called glycocalyx. And what does glyco suggest to you guys? What kind of molecule? Sugars. Again, we can always follow on that. Okay. So, glyco suggests sugar. And again, the capsule being a type of glycocalyx, sugar-based. Slime layers are also a glycocalyx. They're loosely associated, slime layer. If they're densely associated, like what was seen in our picture there, it's very defined around these guys, right? Very defined. This is going to be the capsule, right? Capsules are made of sugars, okay? And proteins as well, sometimes. And they're very sticky. And if I coated you, y'all, when I show you this, the size of the capsule around like the cells here, if I took a people's width, a whole person's width around you, of coating of honey. Same concept, right? Sugar and some proteins in there. Right. If you were covered in that much honey, how could if you're robbing a bank, how is that cop ever going to grab onto you? How is that cop ever going to grab onto you? Right, so that's the same idea. Also, the, why I'm not, so it thinks I'm trying to ask ChatGPT a question and I'm not. All right, so yes, so if you're covered in this, number one, it helps you stick to things. Quite obvious, like you're covered in that much honey, you're easy to stick to things. So it helps with sticking to things. It also helps with building biofilms, which we learned about in chapter 4, is whenever the bacteria live in kind of a community stuck all together, they can send signals to each other via quorum sensing, which is basically just sensing chemical signals. Right? You're pooping, I can smell your poop from over here. They must be eating good though. That's what you might be saying. So that's how the bacteria know how healthy a biofilm is, by sensing those chemical signals. All right, the other thing that it helps with is protecting against phagocytes. Anybody know what a phagocyte is? So is Anastasia here? Okay, so do you know, you want to give me a general, what you think of phagocytes? If it protects from phagocytes, what do you think a phagocyte is?
Yes, cells. Your type of cell.
Mm-hmm. That's fine. We're getting there. They're type of cell, so we'll start there. Okay. So type of cell, if they need protection from the cell, if a capsule is something bacteria have, right? And it protects the bacteria from the phagocytes, which are another type of cell, what type of function do you think that those cells have? Like there'd be one of our cells, right? Okay. So what type of cell do you think it would be in our body? What type of cells would bacteria need protection from? Just general group of cell. Anybody else want to help her out? White blood cells. You're getting there. You were getting there. Okay, we'll give you the credit for that. You were getting there. Okay. So, white blood cells. So your white blood cells go around eating things up. So we kind of talked about that a little bit with our neutrophils, right? That they were able to see endotoxin and say, oh, that's a bad guy. That's an eraser, and erasers don't belong here, right? So those are phagocytes. They see something as foreign, they eat it up. They also eat up cell debris and waste and things like that. So they're constantly just eating up whatever garbage is around, including bacteria and foreign things that don't belong in the body. They recognize it as foreign, they eat it up. So phagocytes, that's what a lot of our white blood cells are, especially neutrophils and macrophages. They're probably our biggest ones. And so we'll talk about that in Unit 3. We'll get into their functions and what they do specifically and how they react to that stuff. But we're getting into the idea of now we have bacteria that we have identified have capsules to defend themselves from being eaten up by our white blood cells. That's a big thing, right? And I'll tell you, most bacteria have a capsule. Most of them do. That's a big thing to be up against. That much honey stuck on somebody. How are our cops going to tackle down the robbers now? They're all covered in honey like that. So they are too. But they have their ways. Don't worry. We'll learn about how our body deals with that. All right, last couple of questions, then we'll get going. When are endospores produced? I'm not going to call on anybody for this. Just go ahead and answer these. Endospores. A lot of people have a tendency to pick one of these that's wrong. You guys did great today with the discussions. Great questions. All right, so our endospores are produced in nutrient deficient conditions. And don't feel bad. Most people tend to lean towards the moist, warm condition. But now I'm going to reframe how you're thinking about this. You're probably just thinking about it kind of off. And what it is our vegetative cells are the active multiplying versions of these cells that make endospores. Vegetative cells are, okay? They multiply, they're the ones making the toxins, they're the ones dividing, they're the ones causing the infection and stuff. But an endospore is just a version of that spore. It's not a reproductive method, okay? One vegetative cell will make one endospore, and one endospore turns into one vegetative cell. The endospore is just a method of hunkering down and waiting out bad conditions. So when you don't have nutrients or when you don't have water, things are bad. It's in a bad way, right? And so they need to hunker down and create endospores. So if you need to revisit that topic in your concept maps, please take the time to do that because we don't have any time to go over that right now. Okay, so you can revisit that if you need to, but that's a process of sporulation and germination. Sporulation, making an endospore, germination, turning back into the vegetative. Making an endospore only takes maybe 3 hours. Going from an endospore to a vegetative cell only takes 1.5 hours. All right, moving on. How would you describe the strepto arrangement? We'll definitely talk about this in lab, so don't stress over it too much. I'm not even going to go over it. Just answer the question. Strepto. Strepto, like strip. What am I doing? Strepto, like strip. Staphylo doesn't sound like strip. Right, so the main groupings, we'll talk about this in lab. I'm not going to go over it too much, but strepto is the chain one. This is our end of our activity. There was so that's the Kahoot. There is an activity for this one, really quick. This is about anthrax, okay? And the organism, it's in here, is Bacillus anthracis. So when we talked about the endospores just a minute ago, there's 2 genera that's supposed to make endospores. So we learned about acid-fast bacteria. Those are the mycobacterium, the bacteria that have mycolic acid, saying acid-fast. All right, we learned about the mycoplasma. Plasma, they don't hold their shape. They don't have a cell wall. Learn about those. The third group that we learned about are our endospore formers. And there are 2 genera that you do need to know that are medically relevant that fall into our endospore formers. Bacillus species, so bacillus will be in their name. And so you automatically know 2 things about them. If bacillus is in their name, they make endospores, because I just told you that. And you also know their shape. They're rod shaped. You know 2 things about them. There's a third thing you know about them automatically. All of our endospore formers, whether they're bacillus species or the other group, which is Clostridium, both of those species are the only ones that make endospores, and they're both gram positive. Are all gram positives able to make endospores? But if you are Clostridium or bacillus, you are gram positive. If you're bacillus, we know you're a rod shape too. And if you're Clostridium or bacillus, we know you make endospores. able to. Those are the only ones that can. And so none of our gram negatives can make endospores. We know that automatically too. If you're gram negative, we know automatically you don't make endospores. So they're both gram positive. They're both medically relevant. This was a story about Bacillus anthracis causing pulmonary anthrax. Somebody that had traveled through an area that was endemic for anthrax. If you don't realize this, then you should probably know that anthrax is an zoonotic disease, which is animal-based. It's very prevalent in livestock. So they're driving through like Wyoming and South Dakota and stuff like that, and they ended up getting anthrax as a result. Endospores can live in the soil and in dry conditions, harsh conditions, and hunker down for decades. So when we talk about somebody having C. diff, which is Clostridium difficile, the endospores are resistant to the antibiotics that we use for other bacteria. They'll just endospore down. And so maintaining treatment for somebody needs to be treated usually for months to get rid of C. diff, because those endospores will hunker down in the gut and wait things out. But if we can allow that bacteria to be stifled down, then our good bacteria can hopefully overgrow. You can have a couple of your endospore forming bacteria, your Clostridium species. You can have a couple sitting in a couple of your stadium seeds. If we have good guys taking up the rest of the stadium seeds, the bad guys won't overgrow and cause C. diff disease. Okay, so you can have a couple. And the idea is not eliminating the C. diff. That's next to impossible. You'll be killing off all the other good bacteria too. We'll get into more of that later. We don't have time for it because we've got lab. I know we went a little bit over. Other days won't be like this. But you guys did great today. If you need to do your makeup questions, then do your makeup questions. You know what to email me with. If you don't, let it go. The lowest score gets dropped. We'll see you guys in lab. We'll head up there. Today we're doing microscopes and stuff. So we'll head to the lab. Anybody heading up there right away, right this second?
The Allen Hospital, that one
So actually, now that you mention it, I know that Congress was, they sent like letters to Congress, like in 2002 or something like that, that had anthrax powder in it.
Yeah, that was short one. NCA.
We can, I mean, yeah.
I watched a documentary on that the other day.
Yes. Yes, that's what I was gonna ask about. I'm just turn it all the way to the right and it'll.
Putting out, they were like making, they like made Teflon and.
Question.